Bom dia a todos. Good morning to you all. I'm so happy and glad to be here today and see you all. My name is Anamaria Camargo, for those that don't know me. I'm an adjunct coordinator at the FAPESP for life science. I'm really pleased to welcome here for the second day of the workshop to celebrate the 20th anniversary of the FAPESP Genome Project. The FAPESP program for genome was a transformative research project. I think we are here really to celebrate the idea. It was carried out by a network of researchers spread over the state of São Paulo and really served as a landmark of genomics in Brazil. So the scientific program of this workshop was jointly built with Marianne Luz, with nice inputs from Professor Luiz Eugênio Melo and Professor Marco Antônio Zago. And this event would not have been possible without the help of all FAPESP staff. So I would like to thank Fernanda, Roberta, Leti, and Claudia Izik for putting everything together, especially the exhibition downstairs that I invite you to visit. It's a nice material about memories of this project, landmarks. So I really invite you for this presentation that is downstairs at the coffee break. I also would like to thank very much our sponsors, Lumina, Eppendorf, and Locus. Thank you very much for the partnership, for the support in organizing this event. And today we'll have the third section of the workshop. We'll have an opening lecture by Dr. Simpson. Then we'll have 3 lectures describing how is genomics today. And at the end, we'll have a special tribute to Professor Ricardo Brentani and an in memoriam lecture that is going to be given by Professor Chi Van Dang. So today we'll celebrate the Human Cancer Genome Project. It was co-funded by the Ludwig Institute and also by FAPESP. So it was also a landmark. We generated over 700,000 ESTs from tumors, some rare tumors, tumors of high incidence here in Brazil. So it was a major achievement. We heard something about that from Professor Perez yesterday. And unfortunately, we will not have time for questions after the lectures, but you can feel free to interact with the speakers during the coffee break. So without further delay, I would like to invite our first speaker, Professor Andrew Simpson, Dr. Andrew Simpson. Andrew Simpson is a biochemist and the Chief Scientific Officer of Orygen Biotechnologia. He was the coordinator of the FAPESP Shilella Genome Program and also the Human Cancer Genome Program. Both were funded by FAPESP, and he also coordinated the Brazilian Genome Network that was funded by CNPq. His aggregating and very inspiring leadership was certainly instrumental for the success of all those initiatives. I had the privilege to be supervised by Dr. Andrew Simpson during my postdoc, and yesterday we heard from Claudia that she was in the right place at the right time, so I was at the right time, at the right place, with the right person as well. And I thank you very much, Andy, for all you did for the Brazilian science. Please.
I should speak in English? Yes. Well, thank you very much, Anna. Very kind introduction. And I think the first time I've been back in this room, 20 years. 20 plus 2 years. Hasn't changed a bit. They may have put in new chairs, but everything else looks the same. Yeah, this is smarter, but the room's the same. And it certainly brings back all kinds of memories. And it's just wonderful to see so many old friends, colleagues here today. I'm sure you've seen this slide yesterday, and I won't dwell on it. But I think it's, actually, I'm going to go back to the slide I wanted to start with. I wanted to start with this slide. This slide, in a sense, says it all. This slide was created to sum up what we did. And what we did was to create a virtual genomics institute. We were virtual when virtual wasn't cool. We were virtual 20 years ahead of our time. And it was a brilliant move. At the time, if you remember, genomics was the thing. Everybody wanted to sequence something. But everybody, except São Paulo, made a mistake of trying to make a genomics institute. That takes time and money and usually political squabbles. By the time we'd finished our genome, everyone else was still trying to build their institutes. So that was the first brilliant thing. And the name here just sums it all up for me. This was a group of very smart people, clever people. This was all about intellect, intelligence. And the name does it. I don't know who remembers TIGER, the Institute for Genomic Research, which was the world's center under Craig Venter for genomic research. And we said, well, we have to have our equivalent of TIGER. And somebody smart said, we'll be ONSA. And someone else said, well, what's ONSA going to stand for? The Organization for Nucleotide Sequencing and Analysis. That was said in an instant. I don't remember who said it, but it epitomizes to me the intellect that went in to all of this creation. And we did the first genome of a plant pathogen, became world-renowned, the FAPESP effort, and we did it under budget and under time. That was pretty good. And that really then opened a whole era, a whole generation of science. I think it made the career, of many of you all, to go on and do brilliant things for decades here in São Paulo. What I want to concentrate on today is the second project that I was involved with, which was the Cancer Genome Project. And the story there was that although at the Ludwig Institute there was support for me being involved in the Xylella project, there were some murmurings at the Ludwig board, and she understands what murmurings at the Ludwig board means. Why is Andrew not working on cancer? Is it because orange juice is good for cancer or something like that? But Professor Brentani says, no. He's just doing that as a forerunner of a cancer genome project. First I knew about it, so we thought, well, what can we do? What can we do as a cancer genome project? And in fact, here, the project was born from some work. I don't know if you remember this, Emmanuel. I hadn't remembered this paper that we did in Renirachu in Belo Horizonte when we were working with schistosomes. When, again, it was the beginning of sequencing, and we wanted to contribute to EST sequencing, express sequence tags, from the schistosome. And at Renirachu, we had a very nice time, but we were in a research environment that was rich in biological variability, lots of specimens of all kinds of parasites and everything, but didn't have any money at all to do research. But we had a PCR machine, and we had a few primers. And what we realized is if you stop thinking of those primers as being specific for a specific organism, lower the temperature of the annealing, they'll bind to anything. in an unpredictable but reproducible way. And you can generate complex patterns of PCR products by low stringency PCR. And we lived off that for years, typing different kinds of organisms and so on. And then when sequencing, express sequence tags came along, we simply applied it to that, to generate small random libraries of ESTs based on primers that had nothing to do with the organism that we were working on. And that worked out very well. And it turned out that from this position of having to come up with something because we weren't able to do what everyone else was doing, it turned out that what we were doing was better. Because ESTs, express sequence tags that we were generating, have two very important characteristics. First of all, they don't come from the ends of the cDNA. Previous to that, people were doing 5' and 3' ESTs. What's the problem with that? The interesting bit's in the middle, the open reading frame. You don't often get there. But our sequences were all derived from the middle. The second thing is populations of cDNAs are very skewed, often to 1 or 2 very highly abundant transcripts, and the rest are all very rare. So you go sequencing over and over and over again albumin or globin or whatever. But generating with these random primers, we highly normalized. So we generated a lot of novel sequence. And our project basically was summarized in these 4 papers, published between 2000 and 2003, remembering that the landmark human genome sequence paper was published in 2001. And what I want to argue today is, looking back at those papers, which I did, obviously preparing for this talk, was a very interesting experience. I don't remember writing those papers. I presume I did, or at least helped to. So it was like reading somebody else's work, and it was very interesting. And I came to the conclusion, not only was it very good work, but it was highly underrated. I think our contribution to the sequencing of the human genome was never really acknowledged. And I'll say why I think that today. So, this is from the first paper. This paper really set out the technology. Emmanuel's the first author. And a lot of the credit for this goes to Emmanuel. This was brilliant work, and if it wasn't for him, I wouldn't have been in Sao Paulo, and we wouldn't have had this project. So I give a shout out to Emmanuel for that. What was really nice is this first figure. I don't know how to do the dot here. Well, that's the problem. Okay, so you can see a smooth curve in the middle. What this curve represents is the percentage of sequences generated by our technology that are predicted to pass through any given point along the length of a standardized cDNA. As you can see, it's a curve, and the most frequent point of sequence is the middle. The dotted data are the actual data that was derived from the first paper. I think it was about 250,000 ESTs, and you can see that the prediction was absolutely spot on. That's where the sequences came from. And that is why it's important. We took a set of genes, fairly long genes, so that we could make the point, and we plotted the 5' ESTs, the 3' ESTs, and in the middle are R ESTs, or STs. And you can see they complement each other. And if you don't have arrestes in the middle, you don't have the interesting bits, the genes. And this is the big problem of normalization. If you have a non-normalized cDNA library and sequence, almost all of your sequences come from common genes. That's at the left. At the right. You can see a long tail of sequences that are coming from what we called at the time unigene clusters. These were sequences submitted to GeneBank, different EST projects, cDNAs, and so on. that are all hitting the same gene. It's called a cluster. And sometimes you've got thousands and thousands of genes. There were complicated techniques for normalizing libraries that have shifted everything a bit to the left. But then if you look at the arrestees, our median cluster size was 125. Normalized libraries were 138, and non-normalized 317. So we were getting rarer genes, and we were getting the best bits of genes. And coming on to the second paper that was published in 2000, and this was really a bioinformatics paper. The first paper was a technology paper. The second paper was a bioinformatics paper. And Sandro, who I don't think is here, was really the bioinformatician who made this possible. And in 2000, as a first step in the whole human genome sequencing, chromosome 21 sequence was published. And so we had a look at what contribution our sequences made to the annotation of chromosome 21. And the point is, I think this is still true today, you can't see human genes just by looking at DNA, because they're broken up into exons and introns. And so we have the Human Genome Project, American government doing these big fragments. You had celebrará doing shotgun sequencing, generating the sequence of the DNA. But the truth is, neither of them, by doing that, were able to find the human genes, because the human genes are broken up into these exons. And if you look at sequence, if you take a small piece of this, the exons can be tiny. There's just no, I don't know if it's still true today, Anna, but certainly at the time, there was no way of being sure what's a gene and what isn't a gene. And this is something I like to show from chromosome 21, where you've got 3 different gene prediction models, Grail, GeneScan, and MZEF, and then experimentally defined, codons, and they don't match up at all. So the only way to find the genes in the DNA is to take the ESTs, or full-length cDNAs, and map one to the other. So it wasn't the Human Genome Project that defined the human genes, it was the cDNA project. And we published our paper, compared it with the sequence that was already published of chromosome 22, not 21 as I said, and we could show that what we did was take the paper as it was published, pull all of our sequences against it. We found 219 new genes. In the time between our paper and the publishing of the first paper, 171 of those had full-length cDNAs. So that proved that our technology was good, but we still had another 48 new genes that we'd found from our own data. And then going on to the third paper, Anamaria is the first author here, and also takes a lot of credit for the huge bulk of work that was done. When we had amassed 700,000 ESTs. And really just want to show here what we showed in the first paper. When you went on to 700,000 sequences, it was still the same story. All of the sequences coming from the middle of genes. And the graphs on the right are really very interesting. At the top, I divided genes into arbitrary classifications of being rarely expressed, poorly expressed, moderately well expressed, or highly expressed. Then I said, how many Orestes had to be generated before we got a hit against each of those genes? So you can see that, and this is all human genes in A. So you can see quite quickly we get to 80% of the highly expressed genes, moderately expressed genes, but the rarely and poorly expressed genes lag behind. But the graphs are the same shape, and that's important. And I interpret the difference in that we didn't sequence all tissues. And most rarely expressed genes are expressed in very few tissues. So we were simply not ever going to sequence those particular genes. And I can make that point by B, because this is just breast tissue. And it's the genes that are expressed in breast, in gene bank, as compared with our ESTs generated from breast. And now you can see that the poorly expressed, the rarely expressed, the moderately expressed, and the highly expressed are all hit at exactly the same frequency as the very highly expressed genes. And that is really quite remarkable. And that took place over, a large project with hundreds of people working, 700,000 contributions to the public databases. And the C is just comparing 3' ESTs, 5' ESTs, and Orestes, comparing with all human genes. And you can see we far outstrip the 3' and 5' ESTs in terms of the identification that we were able to do. And those were the data that were available when the human genome was published. What we did, subsequent to that, we formed an alliance with the NCI, with Bob Strausberg, and put all the sequences he'd done from CGAP and all that we'd done, put them all together and sent them all to GeneBank. And at the time, I think it was something like 50% of all of the human ESTs that come from our combined projects. And that's pretty impressive. I'm talking about the whole world. And this just from the paper summing up the Ludwig FAPESP project and then the National Cancer Institute project. Now, the big question always was, how many human genes are there? There's a very well-known, simple paper by Phil Green in 2001, I think, or 2000, where he had predicted that there are 35,000 human genes. And that was pretty much taken as the number. When the human genome was published, it was said there appeared to be about 30,000 to 40,000 protein coding genes. Our paper says, I won't go through all the lines, but it says, the estimated total genes based on Human Cancer Genome Project and CGAP data, 23,487. If you look in Google today, on Wikipedia, how many human genes are there? 25,000. We got it right, they got it wrong. We should have got the credit for that. We found the genes in the human genome. So, going on from the bulk project, and really I came today really to speak about the science, and say retrospectively what I think we did. What I think we did was more than maybe we gave ourselves credit for at the time. But then we didn't stop there. And a very nice follow-up initiative of Zago and Anamaria was to take all of those ESTs, which weren't necessarily joined up into complete genes, and start building out those genes that weren't defined yet. And again, this was a lot of people working together. These weren't small projects. These were hundreds of people in dozens of labs being coordinated. Fabiana, I can see, is here, who's the first author here. DSC was also very important. Great work over a couple of years, painstaking, where they would take the clusters of ESTs, and here you can see an example. I took this from the paper. On the right is a 3', and you can see several sequences there that come from CGAP. On the left, you can see some Orestes sequences, and there's a gap in the middle. Is that the same gene? Don't know. So what the initiative did was to make primers from the exons, do RT-PCR, and join them up. And here you can see all of the yellow sequences were generated in the project, and join up this gene. And this way, made a note here, I think. there was nearly 60,000 base pairs of new transcribed sequences, 432 exons, and 28 transcripts defined by this project. That was a big deal when the human genome's already been published and they say they found all the genes. And then, of course, that's all summarized there. The final contribution, at least where I was at least peripherally involved, is going on to take one or two genes, because you can sequence the whole genome, you've got thousands, hundreds of thousands, tens of thousands of genes. You can do transcript finishing, you're talking about dozens of genes. But then you start doing biology, you have to talk about one gene at a time. And this was a very nice piece of work. Raphael is the first author, of a type of gene that has historically been very interesting for the Ludwig Institute. And these are proteins that are only expressed in tumors or in the germline, mostly in testes. And these are important because since they're only expressed in an immune-privileged site, the testes, when they're expressed in tumors, they're recognized as antigens. There are antibodies formed against them. So you can use them as the basis of cancer vaccines to treat people with cancer. And they work. And Anamaria found a brand new family of these simply by looking at the RSD sequences. This is one case where she was looking at where things were expressed. And we noticed that these were only expressed in tumors, not expressed in normal tissues. Went on to do a very nice piece of work. You can see at the top there some RT-PCR. The bottom panel are tumors. The top panel are normal tissues. Tissue 1 is testis. All the rest are lung, kidney, etc. So a very specific, what we call CT antigen. And if you look at the bottom right, frequently you can find antibodies against it. So it shows that it's an antigen, immunogenic, and part of our potential arsenal for treating cancer. So I just wanted to go on and say one last thing, which actually comes from another genome project, the National Genome Project, which I was delighted to be invited to participate in, and was great fun, because it actually got me to visit lots of parts of Brazil I'd never been to before, where all the laboratories were. But this project wasn't as well funded as the project in Sao Paulo. And we came to a point where we'd run out of money, but we hadn't run out of sequence. And like any sequencing project, you have gaps. You do your sequencing, then there's gaps. You don't know how big the gap is. You don't know what's in it. This is actually from Shillelagh. We solved the Shillelagh gaps by doing shotgun sequencing. And it worked out pretty quickly and pretty well. But we didn't have enough money in the national project to do that. So actually, we invented a technique for closing gaps in the genome. And again, it comes back to using low stringency PCR, where basically you take a primer specific for the end of one of your contigs, a random primer, do a high stringency, a low stringency, and then just high stringency, nested, and you can build out sequence from the end of your contig. And this we published, and it worked remarkably well, I have to say. It nearly always worked, and we did a nice graph here showing how much you need to generate random sequence before you go on to doing what we call PACE, PCR-assisted contig extension. I made that up, so that's quite fun. And that closed the genome quickly, and if we hadn't come up with that, we would have had a bit of a problem. So that was something that stuck with me. I was very proud of that. I wanted to say today. But what made me really, really proud was the following, that in the Ludwig Institute, we have branches and centers. And one of the centers is in Johns Hopkins, where Bert Vogelstein is the director, who has always been a hero of mine. Ever since I've been in cancer research, he was the man. For me, he invented cancer genetics. He invented oncogenes, tumor suppressor genes, the importance of mutations, and so on. And I had the privilege of going to see him once or twice a year because we were funding his laboratory. there were always highlights, to sit in the same room as the great man and hear what he was working on and so on. And one day I was there, and he said, "Andrew, today we talked about you in our lab meeting." I thought, my goodness, what have I done wrong? Didn't we send the check or something? And it turned out that Bert sequenced the genome once, because he was working with an anaerobic bacteria that his idea was, in the human body, there is no anaerobic space, except in the center of tumors. So if you add an anaerobic bacteria, the only place it can grow is in the middle of the tumor. So you can give it, and it will destroy the tumor. It basically worked. I don't think it's a standard treatment. However, they decided to sequence the genome. And when they came to the point of deciding what to do with the gaps, they looked in the literature and they found our paper and they used it. And there's no higher praise, I suppose, than somebody copying something you did. And when it was your hero, that was my high point, I think, of my scientific career. So thanks, Bert, for that. The paper there, reference 44. So just to finish off, and I hope I'm not too much over time, this is an article in Nature 10 years after the Shillelagh project. And they were saying the Shillelagh was good, well done, but carry on. You need to do more in São Paulo. And the way they were pushing is really in the area of biotech, and saying that ultimately the task of promoting Brazilian biotechnology comes down to the science. It will be up to individual scientists to accept the challenge and expand their research horizons. Well, I took that to heart, really, and I've always loved Brazil. And about 10 years ago today, I got the chance of coming back to Brazil from New York, where I've been the scientific director of the Ludwig Institute, to work for a startup biotech company, Orygen, funded by two major pharmaceutical companies here in Brazil, Biolab and Europharma. And all of you who work here in Brazil know how difficult it is to do that interaction between industry and academia. But I felt that's where I wanted to work. That's what I wanted to have a go at least. So I've been back 10 years, and I've had a go. Ironically, yesterday the sponsors decided they'd had enough, and they're actually deciding not to continue this anymore. So I think I proved that it's almost impossible, but I did my best. And I just wanted to mention the work we're doing, or have been doing there, is also related to genomes, actually. And that is, we're using a structure called poly-IC, coated with lysine and carboxymethyl cellulose, as a stimuli, a non-specific stimuli of the immune response. It binds to toll-like receptor 3, MDA5, and other receptors, and is a mimic of a viral genome. Double-stranded RNA, forms a double helix, and so I'm using genome as a adjuvant. And you can use it in two ways. First of all, you can use it simply to stimulate immune responses. And these are some data that we generated last year here in São Paulo, simply applying the poly-IC Hiltonol to the nose of mice, and then challenging them with COVID. And you can see that it was a lethal challenge. The red line, the black line is the percent survival of the untreated mice, and the red line is the percent survival of the treated mice. It gives you 100% protection, irrespective of the variant, and also irrespective of the virus. It can protect against influenza and other respiratory viruses, and without pathology as well. You can see on the right the percent weight gain. You can see the black, the mice losing weight and dying. On the red, the mice are maintaining at improving their weight. You can also use it as an adjuvant to mix with proteins to make a vaccine. And again, this is where I'm still involved with genomics. This is a collaboration with Ricardo Gazzinelli works in several places, including Sao Paulo, but mostly in Minas Gerais. And he published this paper that just came out in Nature Communications, which is actually brilliant. He showed that if using our adjuvant and his recombinant proteins, he can stimulate full protection against COVID without neutralizing antibody, solely dependent on T cells. And that's very important because T cell epitopes are not variant, they're shared. It was mostly the nuclear protein that was in the vaccine. And so that, to me, is the way forward for COVID vaccines, cell-based. To make it cell-based, you need to have a good adjuvant, like Hiltonol. And if you apply it intranasally, you don't get infected at all. It's not a question of protecting against symptoms, you protect against infection. So it's the way forward. And so we're proud of that. And Ricardo is actually starting a clinical trial this week, funded by the government. So although I've moved on, I've found over and over again in my life, you never really move on. You always stay with science. And it turns out you always stay with genomes. And at this point, I'd just like to say, in my last couple of minutes, big thank you. Thank you for clapping. That gave me a minute to recover. I need to thank FAPESP and Fernando Perez for having the vision and courage to do big projects. You can't do big science without big projects. You can't do big projects without big funding. When you do big projects, you have big successes, but also you can have big failures. And he had the guts to do that. So a great big thanks to FAPESP. A huge thanks to the Ludwig Institute, who came along and didn't say, you can't do this, Andrew. Let's join in. They co-funded the whole project, $5 million, a lot of money then, still a lot of money today with $5 million from. So thanks to Ludwig. Thanks to all the community. Thanks to Brazil. And thanks to my family.
Thank you, Andy. Very hard to speak right now. It was a wonderful talk. And I think you really put into context what we have achieved over the last 20 years. So thank you. Thank you very much for leading us. Thank you. So we'll have the second lecture today, and I'd like to invite Emmanuel Dias Neto. Emmanuel is a biologist. He's a senior scientist at the International Research Center at Camargo Cancer Center. He coordinated the medical genomics laboratory, and he participated and in dimension in the Shigella fastidiosa and in the Cancer Genome Project. At the time, he was a postdoc at the Ludwig Institute. I had also the privilege to share a room with Emmanuel at the time, and we were talking about social media before, and I don't know if he remembered, but we shared a dial-in internet connection, and the blast took at least 3 hours. The blast results took at least 3 hours to come back. So that was amazing what we achieved at that time. So it's my pleasure to invite Emmanuel for this lecture. As mentioned by Andy, he developed the Orestes methodology to characterize the center of human transcripts, and he made a major contribution, and it's a pleasure to have you here, Emmanuel. Thank you.
Thank you all. I'll take this off. Thanks so much, Andy. It was really great to see you and really touched by everything you said and to be here today to see all these good friends in this so special auditorium where so many things happen. Well, the privilege was mine to share the room with Anna. Besides, we're always fighting about the AC because I always liked it. Very cool, and she was not comfortable with that. But we share many other things as well, as you're going to see along these. I even made a little honor here to Anamaria, showing that in 1997, São Paulo was second in the São Paulo State of Soccer Championship. But this is just to recall what I guess Perry said yesterday, that maybe 97 was the year. And there are so many things here, but many other things are not here. And this includes myself doing some PCR work back in René Rachu, as Andy said. This is Álvaro Romanha's lab, which was a great friend that we lost a few years ago. And here, during my presentation, I will name a few people that were absolutely key during this time of my career. And I point here, of course, Andy. He was my supervisor at the master's degree and also at the PhD. Naftali Katz, who was my co-supervisor in the master's, and Sergio Pena, who was the supervisor in the PhD. If it was today, I would not get a grant from FAPESP for a postdoc, because it would be a lot of endogeny, because Andy was also my supervisor in the postdoc. But anyways, at that time, we published a number of papers with very little money. And Andy maybe don't recall, but in the beginning, we didn't even have a PCR machine. We had to go to Sergio Pena's lab in Pampulha, and I didn't have a scholarship. So it was very hard times. I remember that Andy knew some Portuguese, I knew some English, and we were doing some translations of people that would like to publish papers. And that's how we got some money to put food in the car and to go to Pampulha. But anyways, back then, among the papers that we had, we published this little thing in 97 already, so 97 was a good year. And we showed that here we didn't need much RNA. That was something relatively important at that time, and maybe in some circumstances for cancer it's always relevant. But already we managed to show that we avoid the repeated sequence of abundant genes, which back then was, and especially in schistosomes, was very hard to conclude. But then I came to São Paulo, and I remember Andy calling and saying, no, finish this thing and come here. You have to come to the major league. This is where things were happening. And then moved to São Paulo, and a number of people were very, very good and important influences to me back then. I learned a lot with many people. Of course, not all of them are listed here, but we had a terrific time at the Ludwig. Can you imagine having Luisa, having Sandro, having Brentani, Anamaria, Andy, Luis Fernando? It was a boiling pot of, science and challenges and really thinking big. And I remember back in René Rachu, Andy and I used to talk a lot at the end of the days and in the afternoons. And once I remember Andy saying that you can spend the rest of your life working very hard in something that is not important. But if you dedicate your time and your enthusiasm to something that is relevant, eventually you can do something good. So thanks, Sandri, for bringing this which you just mentioned, that we have to do big science. We have to believe ourselves. And sometimes in Brazil, we don't have this encrusted in us, that the capability of thinking that we can do it. So I came to São Paulo, and this I got yesterday from Biblioteca Virtual. And I was so happy to read this. If you go through here, it's very well written. And at that time, you see, Andy's name is not there, but he was the responsible. This is my request for a postdoc scholarship. Back then, I was working at Fiocruz. It was an open position there. I applied and I got the job. But I came and Brentani said, okay, Emmanuel, why don't you apply for a postdoc position? If you get the grant, I promise that I hire you when it's over. So that's what we did. And you see that it started in November 1st, 1997. And it was so fast because I quit my job at Fiocruz in September. We came here, we wrote the project, we sent it, and it was approved in a matter of weeks. So it was really fast, and we managed to implement this. And here we say that all these things marked in red are very well written and very straight to the point. We already knew it. And this was with data that we generated in 95, 96, in a poorly understood organism. And maybe later, as the results in the human genome showed, we managed to prove all these. I've shown this slide. This is the original one that I managed to recover. Back in 96, maybe, no, 97, 98, in a lab meeting that we had at the Ludwig every Wednesday afternoon. And Brentani was sitting just like where Anamaria is. And I thought he was asleep all the time. Every time I look to him, he had his eyes closed. But in the end, he said, Emmanuel, I like that stuff. Please give me your diskette. And it was kind of that big one, because I'm going to Zurich tomorrow, and I'll ask for the patent of this technology. Okay, I saved a copy. I gave to Brentani. He went to Zurich. And after a week, he said, I have good news and bad news. The good news is that they are willing to pay the patent. Maybe Andy will remember that. But the bad news is that they want you to do 10,000 sequences to prove that it's right in breast cancer. And my project, if you recall, it was in colon cancer. It's written somewhere. But anyways, I was working like crazy in the Chilela project and in my spare time. And then we're lucky to have many groups that are working hard and very well. Sergio is one of them, and we invited them to help us. So I'm not going to show this again. Go on and discuss it. And I managed to recover. I was talking to Marie by email, and she said, oh, bring some old stuff, pictures, and etc. So I managed to find very old files. And this is a report, because we send our data to Ludwig to see if they are going to finance it. And they send the data to this guy called Victor Youngenil. None of us knew him. And I remember that Andy said, I would like to play darts with him. Do you remember that? Yeah, because we were angry that eventually he's not going to finance it. But he said, it should be noted that the current collection of sequences was derived from a tissue that has never already been studied extensively, and that more novel sequences may derive from more complex sources. And he mentions before that this can be a very important, significant contribution to define the human genes and to contribute to the human cancer, to the human genome project. So we applied. This is another old thing that we used to have in FAPESP. We decided to apply for a program to finance this, and I applied for the genome of cancer. We got financed, and this is the invitation that I managed to find yesterday for an event that happened in this same auditorium, where we launched the Cancer Genome Project. And I remember that back then, Fernando Reinach was representing the Ministry of Science and Technology, and he said, I'm so proud, because we entered the Chilela project looking for an organism that we have no competitors, that was small and used the most traditional technology. 2 years later, we are challenging the establishment of how to do a human sequencing, how to find the genes. We're using a technique that has been developed in Brazil. We're going for the most competitive area, and we're going for a very big genome. So this was the launch that we had back then, and many good friends, many new people that we start working together, that we still have many collaborations today. This was how we used to have the steering committee agendas, things that we were discussing back then. This is a nice picture taken by Revista FAPESP of the Orestes profiles. Just the professional photographer to capture that. And these were all the centers involved. So I'm sorry I don't have time to go through all of them, but they were key for the success of the project. And this is a report that has been produced by Phil Sharp, which got the Nobel Prize, a researcher of the MIT. Marcelo Bento Soares, a very special guy and a specialist in normalizing libraries, and the same Victor Young-Anil. And it's good to see attention should be paid to the strategic long-term planning of the project, in particular the integration of HCGP sequence data in the analysis of the version of the human genome due in the year 2000. So they already were aware of the potential of the technology that we had. And indeed, as Andy said, when it was published in 2000, we already came very quickly and annotated the genes. And then we start getting, because we deposit everything in the public databases, and people keep writing us. Say, I would like to have this clone. I would like to know more details about this patient. So it was good to see that we are really contributing to advancement of science. And we keep getting these things. Another group asking for information. So usually they write to Andy, and he's passed this to me, and I would go to the source of the library, the RNA, and the aspects of the patients. And these were kind of general things that we had in the routine of the project that maybe none of you saw before. So this was a new RNA preparation from breast, normal breast, that was given to me by Luis Fernando. And we always, after a while, we see if the distribution in the center was still okay, because this was a kind of a way for us to see if there was degradation or some other stuff, and if you're capable of normalizing. So this was the routine of the process that has been produced by Sandro with his tools. And this is to show to Sergio that we also had these reports, how the subgroups that work under him at the Chemistry Institute are producing every now and then. And Sergio sent me this note once. We were in a meeting. I'm not going to show what's inside, but inside there was something that Pérez wants you to write a little piece because he's trying to put in Nature about the schistosome project. And then Sergio asked me, can you help me with that? And we went to a room somewhere. I think this was in Angra in that meeting, yeah. And then we went, and this is the schistosome, so where Orestes started, and I will not say that it ended, but we had a very nice project together that we are not going to talk about today, that Sergio was the coordinator and was published in Nature Genetics. And about old pictures, I managed to find a few. So this was the Jamboree. I'm sorry that I could not find all of them, but I have beautiful pictures that have been taken. So Nagai, Pedro, some other people, I guess it's Sergio back there, I don't know. And this how the kind of strategy that we took to start annotating and making sense of all these human genes. So looking for paralogs, protein domains, expression, skips, focused analysis on colon, head and neck, and other things. So we learned a lot together. So Fernando Lopes Alberto is there, Luisa Tajara, Adriano, I still remember Victor Youngenil, Aline, Betty. So they're very good and important partners. This is me. I wanted to use this shirt. We still have it, but Diana said, no, we're not going to use it. It's okay. Walter Bodman, Sir Walter Bodman, Edna Kimuru, Xin Lu, and Webb Cavaney, Inês, Marines, So, anyways, we published a number of papers, and in the end we managed to publish this one together with Sergio, Eduardo, and some other people that was in cancer research, making the better, most profound annotation of genes expressed in head and neck cancer. I'll just have 5 minutes, I have to move on. So during the analysis, we found very interesting things, and some of them were the presence of some bacteria inside the tumors. And I'm not going to many details with that, but these were there in the paper of the year 2000. We found 135 bacterial sequences among the 10,000. And I remember showing here in FAPESP this to Phil Sharp. And he said, I believe that maybe these are more important than the human genes you guys are finding. And I had this in mind, and I decided to study microorganisms in cancer. Later on, I spent some time in the US. When I came back, I decided to study that. There are many implications in cancer. I don't have much time to go through here. But eventually, we believe that tobacco for head and neck, for instance, and the host genome may contribute and shape the oral microbiome. That in turn is going to metabolize some compounds that are going to, again, cause mutations. And they also play a role in immunomodulation, reduced mucosal immunity. And all that together is going to contribute to cancer. So, as I was not working with Andy Simpson anymore, I found another Andy. So there was Andrew Thomas, which was my student. And he was a brilliant guy. He published that first paper in head and neck. Then he published this one in rectal cancer. And we see that just by looking at the number of bacteria that you have, you can clearly differentiate normal and cancer. Later on, well, this is part of the first paper. You see there's a clear separation of what is cancer, what is not. This brings the possibility of using the microbiome to diagnose these tumors and eventually to intervene. So Andrew published that, then he got a BEPI from FAPESP, he went to Italy, and I don't have much time, but he published this paper in Nature Medicine as a first author, showing that we have a very clear profile of bacteria in cancer. And I have been using this today in my thematic project in gastric cancer. So again, a number of very important people in my lab and in my institution contribute to that. So we look for ancestry of the population. We look for microbiome diversity, how they differentiate cases and controls. And we did more. We wanted to study this together with other people around the world that were developing new techniques. So I joined this consortium in Cornell. Since the beginning, we published a very nice paper last year showing the microbiome of cities, 60 different cities around the world. We found many different and very nice, exciting stuff. This was published in Cell. And some of the tools that we have, and this links everything together again. What's the distribution of H. pylori, for instance, around the world? So we have developed tools that can tell you kind of in heat maps where they are more frequent or less frequent, and many characteristics of the strains that we find. And you can even plot and see the world distribution. You see very strong in Portugal, for instance, which has a very high incidence, the highest in Europe in gastric cancer. Science is also about fun. So we did the Olympion project to see the bugs that were in Rio before, during, and after the Olympic Games. And now we have just repeated that in Tokyo. And my good and great friend Milton Moraes organized that in Rio, which was a big loss that we had a few days ago. And we also did the Monumentum. This to science dissemination to see if you compare the monuments around the world. We would like to do the Christ the Redeemer in Rio, but the Catholic Church is responsible and they didn't allow us to show that there are bugs leaving the statue of Christ. So in Rio, we ended up not doing. These are some of the monuments in the world. In São Paulo, together with Folha, we did at the Ibirapuera. So there are many pictures of myself on the top of this thing and swabbing and collecting. And finally, we did this same technique looking for COVID-19 in Brazil, again with the support of FAPESP. So we took 4 different cities in São Paulo and 3 in Paraná. We developed a number of techniques to evaluate the distribution. And this is what we have. We're just preparing the manuscript. We have to discuss this later. Luiz Eugênio has very interesting data here. I just mentioned to Luisa, who was a strong supporter. These are the distribution of strains that we find in healthy people, people that are using the subway, people that are arriving in the airport. And there's a lot of virus there, at least a lot of RNA, and these may have very important consequences for, in real time, allowing us to see where the sequences or where the virus is spreading, how is it spreading. And maybe there are some correlations with death curves. You see that Delta caused a lot of deaths in the world, but not in Brazil. And we believe that these spread of variants may help explaining that. And we also manage to map a number of variants that we find in the spike protein just by using these genomic data in large scale. That's it. If there's one person that would like to dedicate this conference that I gave today is Dr. Brentani. Since the beginning, as I have said, he said that, come, I want you, I will support you, and he was absolutely key for me having my lab today. And also, Andy is another key person in that. Since the beginning, I remember I was looking for, I don't know what to do after I graduated, and Naftali said, there's a gringo here. You have to meet him. You have to work with this guy. And then I was planning to go abroad and to do some stuff, but why not? Why not work together with this brilliant guy? We also had an extreme synchrony and very good times working together. So thank you so much for that, Andy. And at last but not least, thank you, FAPESP. FAPESP was the major supporter of all of that and the major contribution for science in Brazil. And thank you so much for your attention today.
Thank you very much, Manuel, for the good memories, nice pictures. It was an amazing time, and I think you put into context what we achieved and how it's impacting science today. So thank you very much. So our next speaker is Sergio Verjovsky. Sergio is a medical doctor. He is currently the leader of the Cell Cycle Laboratory at Butantan Institute in São Paulo. He joined the Shigella fastidiosa Genome Project and the Human Genome Project, as he was a full professor at the Department of Biochemistry at the University of São Paulo. And he coordinated one of the sequencing laboratories of the ONSA and also the Schistosoma mansoni EST project. Thank you, Sergio, for joining us today. Thank you.
Well, I thank very much the organizers for giving me the opportunity to speak to this select and beautiful audience. Well, I'm trying to have a timer here. Okay, yeah. I'd like to start talking about long non-coding RNAs in cancer by showing a survey that I just did on the number of papers in PubMed that have been published And you see that the first ones appear in 1976. And this is almost 50 years ago. In the 2 decades that span up to 2000, which is the year we are concentrating onwards, there were about 20 papers on long non-coding RNAs per year. And actually, the subject came up to attention when the group of Willard at Stanford published the EXIST long non-coding RNA, which is an inactive X-specific transcript that modulates imprinting of the X chromosome. And then attention started to be drawn to the long non-coding RNAs, and there were about 120 papers on long non-coding RNAs per year in the world, which kept around that number up to 2010, when really high-throughput sequencing started showing the loci of transcription of non-coding RNAs. And as Andrew pointed, the genome was published in 2001, and it helped localize this mysterious transcription that occurred outside of the protein coding genes. As for long non-coding RNAs in cancer, there were about 40 papers per year at the end of the year 2000. That's about 1/3 of the long non-coding RNA papers. After 2000, we got a sharp increasing after 2010, and about 1/3 of the papers on long non-coding RNA today are on cancer. And where were we? This was the time between 2000 and 2005. We're here. You brilliantly heard Andrew showing, and this is one standard output of our project that, and they use here the nomenclature that was at the time. Those 1,000, where is that? At the bottom there, 1,163 ESTs were produced, mapped, and known human genes and unigene contigs were detected in our project. But there was about 30% what we call no matches. So what were those things? By sequencing cancer, we thought we were discovering new cancer genes, or perhaps new cancer gene exons. And these are the 3 papers that have been extensively discussed there that contribute to that. And so at that point, our lab, we thought maybe we should explore that. And so we decided to take those clones. We selected about 2,000 clones of the so-called no matches, and another 2,000 clones of protein coding genes of interest in cancer, and built those arrays. And I said, well, maybe if we look at a cancer like prostate cancer and try and find non-coding or possible new exons or new genes that will correlate with the degree of malignancy of the prostate cancer, we could find some significant new genes. And it turned out that 24 out of the 56 genes which we pointed by their protein coding numbers were not protein coding genes. They were non-coding RNAs that I gave Eduardo Reis, which is the first author in my lab, was the postdoc, and the task to prove that there were new exons of the genes by doing PCR with the exons in the vicinity. And he came to my desk every week or so. I failed. They don't work. Then I said, a challenge. What if we bet that they are on the opposite strand? And then he could confirm that they were all in those loci, but they were antisense genes. And so we wrote a paper, and I came to Brentani and Emmanuel, and I briefly gave a spoil to Manuel there. Do you remember? And he said, oh, I remember that. Yeah, I'm going to talk about that. I remember that meeting that I showed Brentani and Emmanuel the paper, and I said, wow, but this is so strange. They're not new exons. They look antisense. And Brentani raised from his chair, reached those metal files that we had printed copies of the paper and said, oh, there's a paper in 1986, 6 years earlier, that show antisense transcription on the CMYC oncogene. So give more emphasis to this. And so we changed the title. We pointed to antisense as a really new thing, and it said, send it to Nature. Ought to be published in Nature. So we sent there, and it wasn't not accepted. It came back, well, at this point, everyone knows there are non-coding RNAs. You must show a mechanism. It turns out that 20 years later, there's still no mechanism for those things, but it ended up in oncogene. And I came back to the paper. There's only modest thanking at the end of the paper to Emmanuel and Brentani for the discussions and writing. But that's the big story there. And then by the end, by 2007, it apparently became more evident that identification of the eukaryotic mammalian transcriptome complexity has redefined the concept of a gene. So there were protein-coding genes in red at the top there, right? But there were also long intronic non-coding RNAs outside of the loci. In the protein-coding loci here, you have intronic non-coding RNAs, antisense non-coding RNAs, and it became evident that non-coding RNAs were a large part, much larger than the protein coding. And that's where the wrong numbers came. It was thought that there were not only 20,000, but 30, 40, 50. There was even an Israeli company that had a secret database that they claimed there were 100,000 genes. And when the mapping of the transcripts became more clear, and that the rest was probably just long non-coding RNAs that had the function. At that time, we also were brave enough to be competing with the ENCODE in 2007, and Elder Nakaya mapped what was already there. And remember that Andrew pointed, we had contributed to half of the information on the database. At that time, to produce 1 million reads, it took 100 people, years. Now we produce 300 million reads in 2 or 3 days, in one run. But it was a really a big thing, and we thought we could explore. So I gave the text for Elder to map, and we did a genome mapping and expression and analysis of intronic non-coding RNAs that were in the public domain. And we set up to design a custom oligomicroarray to order through Agilent. And we found patterns of expression of different non-coding RNAs in prostate, kidney, liver. And it turned out that the most highly expressed intronic long non-coding RNAs mapped to loci of protein-coding genes related to regulation of transcription. So at that time, we pointed the fact that possibly are part of the regulation of gene transcription. And then after more than a decade of studies of long non-coding RNAs, there are examples of mechanisms of long non-coding RNAs associated with cancer, like epigenetic silencing non-coding RNA binding to PRC2 complex and regulating the transcription of genes. There are spliced variants produced by an example, MALAT1, that binds to the splice regulate the protein and regulate splice variants. There are examples of loop of like that Hulk non-coding RNA that drives, that binds a microRNA that regulates the protein kinase that regulates its own expression. And there is also modulation of protein activity, examples of important cancer non-coding RNAs. And at this point, we know that long non-coding RNAs are really associated with all hallmarks of cancer. And these are 50 non-coding RNAs. And they were reviewed in 2016, there is about another 50 that are characterized, but not more than that, whereas there are almost 100,000 non-coding RNAs in the human genome, and as Andy mentioned, have to work one by one, probably, in the future. And I modestly marked NRAS antisense non-coding RNA in the RAS1 gene, which is a contribution of my group, and I'll come to that. So I'll briefly show you now NRAS1. NRAS1 is a non-coding RNA inhibitor of a tumor suppressor gene RAS1A, and that causes increase in cell proliferation. Hipster is a regular long non-coding involved in developmental program. I'll talk about androgen receptor-associated link RNAs, and PVT1 link RNA, which is one of them, is an oncogene that's associated with androgen receptor, and signals a transcriptional repressive program of tumor suppressor protein coding genes. So the RAS1 we had picked in 2003 in Eduardo's panel there. RAS1A was the most increased antisense non-coding RNA that was increased in that locus. So it took us 9 years to, work the function of that. So we extended by race. We showed that it really is antisense of this important tumor suppressor gene that is downregulated. And so because it is overexpressed in cancer, in the locus where this tumor suppressor is downregulated, we we named it NRAS1, and we raised the hypothesis that maybe it is binding a repressor complex. And indeed, we showed that NRAS interacts with PRC2 complex, recruits PRC2 histone modulator to the locus, specific locus of TF of RAS1A, not the genes in the vicinity. And we showed that the NRAS overexpression inhibits tumor suppressor RAS1A and increases cell proliferation. And then we found another interesting non-coding RNA in the locus of TFP2A, which is a transcription factor known to be involved in various cancers, including prostate cancer, where it is downregulated. So again, we thought maybe this one is another example. Let's see if it binds the polyclonal repressor. And in fact, it quite acts differently. It doesn't affect any of the TFP2A isoforms, and knockdown of this non-coding RNA showed that 380 genes were differentially expressed, and 99% of them were upregulated, and most of them were involved in self-differentiation. So we thought this is an interesting gene that could be explored. Then we wanted to continue working on prostate, so we wanted to know how many link RNAs are there, and we found 3,900 new link RNAs by doing deep sequencing of prostate, and then we thought maybe some of them bind to androgen receptor, and ended up that androgen receptor bound over 600 long non-coding RNAs. So it's an RNA binding protein, and we call them androgen receptor associated ARA link RNAs, and they are closer to the neighbor protein coding genes, the long non-coding that don't bind androgen receptor. The genes in the vicinity have a set of important functions, including embryonic placental development, RNA processing, and the chromatin topological associating domains of the loci where the ARA link RNAs bind act as an enhancer because of the marks that we found enriched there, particularly the K4 monomethylated and the K27 acetylated. The highest the mark, the highest the correlation with the protein coding gene in the vicinity. So link RNAs were identified to interact with androgen receptor. They're candidates to regulate the expression of the genes in the vicinity or in the TADs, and they are candidates to be further studied on protein, non-coding RNAs. The last example is PVT1, which was one of the link RNAs bound to androgen receptor. And we thought maybe this regulates the genes that are, because PVT1 was shown to bind in HeLa cells polychrome repressor, we thought maybe in prostate they would do the same. And we first confirmed that androgen addition to a prostate cancer cell culture inhibits, on the upper left, not only activate some genes, but also inhibit a set of over 1,000 genes. And when knocking down in the lower part here, PVT1, genes were upregulated. And by combining the ones that were inhibited with androgen and upregulated when PVT1 was knocked down, we find 160 of them that overlap. And those were candidates to be inhibited by PVT1 and polychrome repressor. And it happens that those genes are enriched in a set of tumor suppressor functions, which tells that PVT1 might be inhibiting a lot of tumor suppressors and be their cells apoptotic functions being repressed. Those set of genes also predict high-risk cancer in the CGA database, and so we finally showed that repressed set of genes enriched in tumor suppression functions may lie behind the aggressive phenotype of tumors in PVT1. Now, what is the future of that? In the past 5 years, I also surveyed the publications, and in the beginning was only my lab here in São Paulo. Now, in the past 5 or 6 years, there's an average of 15 papers per year on long non-coded RNAs in cancer in Brazil. More than half of them are from São Paulo. And there's a big interest all over the world in long-coded RNAs in cancer. But are they susceptible to be targets for treatment? And, well, I believe that targeting them with antisense oligonucleotides or siRNAs, which have been approved as FDA drugs for test in other diseases like amyloidosis or chylomicro anemia or hypercholesterolemia, they will probably be targets, non-coding RNAs in cancer. But for that, you have to sort problems of specificity, of delivery, and eventually, if we understand how they bind specifically to some proteins and modulate them, we could find the small molecules that would interfere with non-coding RNAs and proteins. So I think there's a brilliant future for that, but I didn't dare to specify what will be the outcome for treatment. So with that, I finish my talk thanking all the people in my lab, but especially thanking Andrew Simpson and dedicating this to Brentani. Both of them were very special, critical people, allowing my group and others to expand what had been produced by the group that originated and worked at the beginning of the project. So I'm very happy to be here and find all of them, and I thank you very much.
Thank you very much, Sergio. I remember those no-match Orestes. They were a question mark at the beginning, and it's so nice to see the story turning out. And also, I think Andy mentioned that Orestes helped to identify protein coding genes, but it also did to uncover a new world of non-coding RNAs. So thank you. It was an amazing talk. So the last speaker of this session is Dr. Rui Reis. Dr. Rui Reis is a biologist, the scientific director of do Instituto de Ensino e Pesquisa do Hospital do Câncer de Barreto, and he's also an associate professor of human genetics at the medical faculty, the Minho University in Braga. Rui has been working in cancer genetics and genomics since 1996, and it's a pleasure to welcome him here today. He's currently the leader in cancer diagnosis and biomarkers. Thank you, Rui, for accepting the invitation.
Bom dia, good morning. So, I want to thank the organizers, Marianne for the invitation and Maria for the introduction. And it's really a pleasure to be here. I'm really proud. It's been a privilege to, during these 2 days, witness this fantastic endeavor that was the FAPESP genome program and fantastic science that is being done. So I'd like to tell you that I was surprised and humbled by the invitation because 20 years ago, to doing molecular research and genetics at Barretos was only in our dreams. So we are a very newcomer, and I hope to show you that our story is also a legacy of what FAPESP and this project was meant. So first of all, so probably most of you know, but Barretos Cancer Hospital is a private non-profit organization. We attend only a public health system. And our core basis is to treat everybody with care and compassion. So that's why the name changed recently to Hospital de Amor. And we are focused on underserved and remote populations. So based on this philosophy, the hospital that is located in a very small city in the rural area attracts patients from all over Brazil and now is a very big institution. And to cope and to overcome the problem of the distance between patients and their family, the hospital has spread around Brazil. And you can see here in these pink dots are our facilities of cancer prevention and cancer treatment. So the hospital has also a big history of mobile units to reach the patients in their places. And usually around average we have 14,000 new cases of cancer per year. So it's currently one of the biggest cancer institutions in Brazil. So our story on research is much recent. So I arrived to Brazil, to Barretos in 2010, to the inauguration of the Oncology Center, Research Center. And it was myself plus 4 young PhD, young postdocs. So the hospital has no history. DNA isolation was not known. And we have an empty lab. So outside a big academic center. So it was a huge endeavor to start from scratch and to arrive to our goals, to our mentors that were here at São Paulo and many of the collaborators that are present here. So since cancer is a genetic disease, so our main focus was genomics, but we want also to not only to find alterations, but understand their function. So we are also state-of-the-art labs for functional studies, like in vitro and in vivo models. So I said that we started from the scratch, but was not really that story because to do genomics we need sample of patients. And the hospital had already an institutional tumor bank that started in 2006. And started in 2006 after a visit of Professor Brentani, that went to the hospital and told to our CEO at that time, so you should have a tumor bank, we have a lot of patients. And the answer was, but we don't have money, we are not thinking doing research. This is for high league, only for São Paulo institutions, for USP. They no, do it, build the tumor bank. I'm sure that a lot of good things will come in the future. So Brentani was a key person in the research that we have currently. So it's an institutional tumor bank, so all the patients that signed consent, we collect blood, tumor tissue, clinical data. So currently we have approximately 300,000 samples from 50,000 patients. And here we have, so the distribution, since we have patients from different parts of Brazil, the studies that we perform there can have a glimpse of what is going on the country. However, as you can see, and it's well known, most of biobanks are in Sudeste populations, so we don't have really a full picture. And thanks to a Pronom grant that will start next year, in our units, we'll have new 3 biobankings in these remote areas. So the idea is to have a more complete picture of what is going on. And of course, with this tumor bank, we started several genomic projects at our place, and especially with collaborations and in consortium. So one of the biggest and first collaboration that we started was the TCGA that all of you know. So it's a landmark study to analyze tumor at different omic platforms. So we participated as a tissue source, so you were sending samples and clinical data, and we participate in the work discussions of the results. And this for us was really a huge learning process, how all these study come along. We are not the only Brazilian participants. I think USP also participate with some cancers, but in the end, TCGA was mainly American population. And this is currently a big problem because more than 95% of genomic data available of the patients, they came from European or North American population. As you can see here, South America is a very teeny part, and especially the mixed population like Brazilian is even less represented. So there's really a huge need to understand and to the genetics of Brazilian population. So with that, we started some projects at Barretos, and I would say that in 2015, so probably is one of the biggest initiatives. So we are doing both sporadic tumors and hereditary. And in sporadic, so you can see here, so we are doing whole genome sequencing of melanomas and project of whole exome sequencing of different type of tumors like melanoma, like lung, breast, and others. And at germline level, we are doing whole exome sequence of hereditary cancer of patients that don't have mutations on the classic BRCA1, BRCA2, or P53. So to identify novel genes. And due to the sake of time, I will only focus today on the work that we have been doing in melanomas. So, as mentioned, so all of projects we are doing pair sequencing. So tumor, DNA from the tumor and DNA from germline. So we did the whole genome sequence at Mendelix here in São Paulo. We did for all of them target sequencing to validate the most important genes. And then the bioinformatics was done at Barretos, and with a strong collaboration with MD Anderson that we have proud to be a sister institution. So this was a collaborative study. And since we were doing these projects and with that magnitude, so we were able to be part of the International Cancer Genome Consortium, and it was the initiative from Brazil for skin cancer. And we also entered there because at that moment, the ICGC concerning melanoma only have patients from United States and from Australia. So there was a big gap and need for other ethnicity populations. So one of the first things is to understand what was, so there's a lot of mutations and mutation burden. And we identify more than almost 19,000 mutations. So from a group that was just starting, this was a huge amount of alterations. And this goes well with what was already published from the Australian and the States American cohort, that melanomas, they are driven by UV, so, and they have a lot of mutations, so are the one of most higher mutation burden. Another thing really interesting that we can learn from the genomics is the mutation signature. So we know that each mutation, the substitution of a base to another base, a nucleotide, is linked to the type of agent that induced that mutations. And knowing the context, we can now have what is called the mutation signature. So depending on the type of signature, we can deduce what was the carcinogenic agent that induced that alteration. So we identified in our samples this signature 7 that is associated with UV-related mutations. And when we applied to all our cases, we found that different from the other populations, not that more than 90% are UV signature related, we found approximately 50% and the others with non-UV signature. So it means there are another etiological factors ongoing in our cases. And of course, we want to know what are the driver genes that are present. And one fact that is really different from our population to the other is not only the patients that are Brazilians, but the histology. So you can have mainly two types of melanomas. It's a heterogeneous group, but mainly two types. The cutaneous one that arrive on skin, and acral melanomas that occurs in the palm of the hands and feet, and are more common in darker skinned persons. So we have, that was a darker brown, so we have around 30% of our patients from this histological subtype. And as you can see here, so there's tumors with a high mutation burden, even higher, so that are on the top. We have this primary or metastatic. And then these are the top melanoma driver genes that we were identified. And the TCGA, based on this, classified the tumors in 4 molecular subtypes. The BRAF subtype that has mutation on the important BRAF oncogene, so we identified around 56% of cases. Sorry. So the most codon alterated was codon 600. So this was already known, but it's really important for the ones that are not from the field, because currently we have target therapies for these mutations, and in fact they are being using, and in our clinical context we are performing the mutation and the patients are treated if they have this mutation. So this is a clear example of how genomics affect the daily life of the patients. The other subgroup identified was the RAS subtype that has mutations on the NRAS, KRAS, GRAS, the NF1 subtype, and the triple negative. So the triple negative is really rare in other populations. We found very frequent here. And you can understand the triple negative, so don't have mutations on the classical. So it means that we don't have even probably target therapy, so there's a big interest in this group. So we compared our population with others, so there's a lot of similarities. What really struck us was the NRAS frequency that is really below half of what is described, so there's really something different going on in our population. Other analysis that we can, you can extract is the chromosomal alteration, so we identified very important regions that are associated with losses, like at chromosome 9 and 10 with the P10, so the important and well-known tumor suppressor genes, amplification regions where oncogenes are driver of these events, and the structural aberrations that fusions between chromosome 9 and 12 as found here. And we can integrate all this data and understand the pathways that is alterated. So 53 and RB are present in the great majority of cases. And we found a lot of alterations in telomeric maintenance and methylation. And methylation here I can bring to another history, that is, methylation is not only important for tumorigenesis, but can use as a molecular sensor of the risk factors that were involved. And since UV is a main risk factor, so you want to understand better what was this proposed. So we integrate this genomic data with epigenetic data. So in collaboration with International Agency for Research on Cancer. So we analyze cutaneous melanomas from our patients. We compare with American subtypes to try to identify what are the methylation markers that are important in these cutaneous and associated with UV, non-UV exposure, and then analyze our cohort of acromelanomas. As I mentioned, they are more frequent in darker skin and really poorly explored. So we want to understand if there's any epigenetic markers. So just some features of the population. So as expected, cutaneous melanomas are more in white people, compared with pigmented skin that found in acro. UV signature that is present in the majority of cutaneous is not so frequent or is less frequent in acro, so there's another driver event. And then we compare by the molecular subgroups. BRAF is much more present in the cutaneous, and there's several features that are really showing that are molecularly at different diseases. So we perform the methylation profiling of these samples in cutaneous melanomas, and we identified two big groups, the cluster 1 and cluster 2, that were different from the other cutaneous, and we identified these clusters, they were non-UV related. So methylation profile shows there's a different genes involved. Most of these genes are involved in immunological functions, so this is really important in context of immunotherapy and also in some skin diseases. Then we want to understand what were the major regions, differential methylated regions among these UV and non-UV related. We identified several CPG islands, and in the bottom down, we identified 3 genes that were really distinct between in the same white people that are UV related and non-UV related. And the top gene was TAP-BP, that is a gene that is not well studied. So we validated by bisulfite methylation. We showed that methylation status was associated with expression. And more importantly, the methylation of this new gene can be associated with a different prognosis on the patients. And finally, we compare then the cutaneous with acro. And interesting, we found that the cutaneous melanomas that don't have the UV signature, they are similar, more similar to acromelanomas rather to cutaneous. So the epigenetic mechanism is more driven than the places where the tumor is occurring. And also we are analyzing what are the main pathways and genes. So this is a glimpse of the history that I like to show you. And recently I found this picture, and I think genomic is that. So usually we are focused on generating the data. And in fact, something that I learned from ICGC, and it's very different from Brazilian funding agencies. In ICGC, when you apply for a grant, for example, a sequencing grant, if you put 1 million for generating the data, you put 3 million for analyzing the data. And usually, when we apply for a grant, we put 1 million for sequencing and just some peanuts for the analysis. But it should be the other way around, because it's much more difficult to build, to sort it, to arrange, to process virtually, and to have a story. For the story, you need good clinical data, and we hope that doing that and genomic will change the history of melanoma and cancer, becoming a more tolerable and potential curable disease. And of course, to build this nice story is a huge amount of persons and time and money. And I want to finalize, thank you. First and upmost, Barretos Cancer Hospital, all the team, our CEO, Henrique Prata, that is a farmer, but like many visionaries, he sees what is the future, and the persons that were directly involved. So Adriana Evangelista, so she was the leading bioinformaticians. Ana Vicente, it was a PhD student that with a FAPESP fellowship were able to be in France and did this beautiful methylation study. Marcia, that is responsible for the biobank. André Carvalho, that was our former director, the one that also brings this idea of bringing molecular oncology to Barretos, and Vinicius, that is leading of the melanoma group. Of course, with a lot of collaborations, I just put here the ones that are more crucial. So MD Anderson and Futuro, where Adriana spent some period and learned a lot of the bioinformaticians that were applied. My PhD alma mater, IARC, and my friends Zengo and Akran, that were really instrumental for the methylation. Mendelix, João Kitajima. So as you can see here, Barretos is really a legacy of the FAPESP, not directly, but indirectly. Most of our persons were trained with someone that was involved in the past, so it's a second generation of a successful story. And I like also to thank you, Professor Ricardo Brentani. I never met him when I arrived in 2010. I was in São Paulo, so I didn't have the privilege, but indirectly I know that he was really a key person in having the infrastructure that was basis for starting the research. And many of our André and Vinicius, they came from Camargo, so they were embedded with that scientific spirit that they brought to Barretos. And finally, of course, the funding agency, FAPESP, mainly on grants for our students and also projects, MPT, Pronom, and FINEP, and thank you very much for this great opportunity to be here with these outstanding. I'm really humbled to be here in front of all these big names in Brazilian science, so thank you very much.
Thank you very much, Rui. I think the idea was really that, to bring the next wave of genomics in Brazil. So thank you for putting genomics into the clinical context and also for highlighting the need of increasing diversity in genomic studies. I think that's key. So we'll have a break now. We have a coffee break downstairs. Visit our exhibition. We should be back 11:30 for the lecture, the final lecture of this session. Thank you very much. Oh, please, don't leave your personal belongings here. We are going to lock the auditorium, but we will not be responsible for leaving bags and other stuff here. Thank you.
Take your seats. Welcome back. It's a pleasure to invite Professor Marco Antônio Zago, President of FAPESP, to the tribute to Professor Ricardo Renzo Brentani.
Before starting, I wish to introduce a different thing here. And I will speak this part in Portuguese. Nós recebemos cumprimentos por esta atividade, e uma delas, bastante valiosa, a meu ver, vem de um grupo do Paraná, representado pelo superintendente de desenvolvimento tecnológico do Estado, Aldo Bona, o Ramiro Verjovsky, que é o diretor científico ou presidente da Fundação Araucária, o David Figueiredo, que comanda o projeto Vale do Genoma, o Grupo Jacto e a Fundação Sunji Nishimura. They send us this compliment for the 60 years of FAPESP and 20 years of genomics in Brazil. So I wish to thank them for this.
Thank you, Professor Zago. To this ceremony, I would like to invite Professor Marco Antônio Zago, the President of FAPESP. Also inviting Dr. Chi Van Dang, Scientific Director of Ludwig Cancer Research. Dr. Paulo Hoff, General Director of the Cancer Institute of the State of São Paulo, ICESP. Dr. Giovanni Cerri from the Medical School of the University of São Paulo. Professor Carlos Pacheco, the CEO of FAPESP. Professor Zago, you have the floor.
Okay, good morning, everyone, my friends, Professor Luiz Melo, our scientific director, accompanies us here. And as part of the commemoration of the 60th anniversary of FAPESP and 20 years of genomics research in Brazil, FAPESP and I personally are very glad to introduce this tribute to Professor Ricardo Renzo Brentani, who was the director of the São Paulo branch of the Ludwig Institute for Cancer Research. He was also the first full professor of oncology of the University of São Paulo, the director of the AC Camargo Cancer Hospital, and the president of the executive board of FAPESP. And this is why these people are here. For this reason, we invited to this table Professor Chi Van Dang, present scientific director of the Ludwig Institute, the president of the executive board of FAPESP, Professor Carlos Américo Pacheco, Professor Giovanni Guido Cerri, director of INRAD, the academic institute that incorporates the original chair of oncology, and Professor Paulo Hoff, who is the director of the Cancer Institute here in São Paulo, and who is the inheritor of the chair. He is the present professor of oncology at the University of São Paulo. So, Ricardo Brentani graduated in medicine at the University of São Paulo in 1962. His doctoral thesis, completed in 1966, described the processing and properties of the nuclear RNA and pointed out to the role of a nucleoli. His, you changed it? Okay. His supervisor was Isaias Rao, one of the leading scientists at the time, whose impact in Brazilian science is well known by everyone. As Isaias Rao says, it took more than 15 years before the importance of the nucleolus in the synthesis of messenger RNA was recognized. Thus, Brentani started his science career in the company of researchers who were working at the frontiers of biochemistry. In addition to Isaias Rao, his co-authors in 4 articles published in Nature at those initial years were Thomas Mack, Moisés Rabinovich, and Maria Mitzi Brentani, who became his wife and long-life collaborator. As a young student, he had an independent mind. In the words of one of his supervisors, Moisés Rabinovich, he didn't ask before he did things. As an example, he refers to us the exchange of correspondence between Brentani and the philosopher Karl Popper about a point of disagreement in one of the philosopher's newly published book. Along his scientific life, he has published more than 300 articles. In addition to the role of nucleoli, he dedicated his attention to the characterization of messenger RNA and later to questions related to collagen, cell adhesion, and metastasis, as indicated in this publication from 1985. It published this paper in Science showing that Staphylococcus aureus, which invades tissues and causes abscesses, has laminin receptors, whereas Staphylococcus epidermidis, which is not invasive, does not have the laminin receptor. As he and others had already demonstrated that laminin is involved in cancer metastasis, he suggests that the prokaryotic and the eukaryotic cells might use similar or the same mechanisms for tissue mutation. In his final years as a researcher, he was intrigued by the physiological role of prions, those isoforms of infectious prions that are involved in the origin of such diseases as the transmissible spongiform encephalitis or medical disease. Thus, he always worked in the fields of molecular and cell biology, although these denominations were applied to this area of research only more recently. He was praised for his sharp intelligence, his practical sense, and capacity to take difficult decisions. Those features were essential in his life as scientist, as well as a director of an important cancer hospital and research center. But one important aspect of his complex personality, not known to everyone, was how he treated in earnest the ethics of research. He never accepted, for instance, to be one of the authors of the Xylella paper, neither to receive the medal awarded by the state government to all researchers who participated in the project. All who took part in the project, as men who are here today and myself, remember very well his role in the Xylella project and the Cancer Genome project extended far beyond that of a financial supporter or manager. Brentani will be always remembered because he was one of those rare persons who managed to be at the same time successful as scientist, as a science policymaker, and as the director of the largest cancer hospital in São Paulo at his time. He was the first director of the Ludwig Institute for Cancer Research in São Paulo. From the beginning of its activity in 1986 until his death. He was substituted by Luisa Villa, who is there, who were the head of the virology research group. The institute maintained a branch in São Paulo for 30 years. In his 2003 report on the scientific activities of the branch in São Paulo, Brentani listed the following research groups: cell and molecular biology, cancer genetics, computational biology, molecular immunology, virology, and the laboratory of molecular biology and genomics. You already saw this, but this is a different way to put it, Manuel. The São Paulo branch's expertise in genomics became internationally recognized because of the Orestes approach to identify express the sequence tags and the transcript finishing initiative, both collaborative projects funded in partnership with FAPESP. Around the turn of the century, the Cancer Genome Initiative of Ludwig and FAPESP was one of the most important individual sources of information about expressed human genes in the world, as it was already stressed in the beginning of this day. In 1990, he became the director of the AC Camargo Hospital, known as Câncer Hospital. Since that moment, the hospital became an international reference center in research, teaching, and assistance in cancer. Following his view, as he pointed out, I thought that the hospital should be more than a hospital. It had to become a teaching and a research center. As a consequence, since 1996, the hospital became the only private-owned hospital to maintain a successful postgraduate training program. An excellent administrator, he also balanced the finances of the hospital, as Isaias Hall again commented. Brentani managed to put on foot an institution that was bankrupt, Câncer Hospital. He was member of the board of trustees of FAPESP in 2004, when he was elected to the position of president of the executive board, position that he retained until his death in 2011. In the opinion of Celso Laffer, former president of FAPESP, Brentani, with his strong personality and sharp intelligence, made an invaluable contribution to raising FAPESP's level of quality. This was not surprising, he was once more reprising the pattern of his life in any enterprise that he undertook, bold, courageous, innovative, looking for the highest quality. But never lacking in good humor, and never missing the opportunity for a joke or a good laughter. Perhaps his more enduring legacy derives from the position that he had as director of the Ludwig Institute for Cancer Research. This made it possible for him to link together the two institutions, Ludwig and FAPESP, in one of the programs that had the biggest impact in the 60 years of life of FAPESP. The Genome Project, as it became known, changed the scientific panorama of Brazil. Brazilian biotechnology has matured to the point at which its scientists are now players on the international stage. That's what Nature said. The skills and knowledge that we started to acquire at that moment are highly valuable today. The legacy of that period extends to all fields of research of life sciences. As for instance, in our ability of a large-scale fast sequence genomes that demonstrated to be crucial during the pandemics of COVID, or when our university hospital in Ribeirão Preto is the only place in Latin America that is performing genomic manipulation necessary to treat cancer patients with CAR T-cell therapy. For many years, I had the privilege to work closely with Ricardo, to enjoy his friendship, and sometimes also, as everyone near him, also his oppositions. I am very honored and very glad to have had the opportunity to deliver this tribute to him today. Thank you. And they told me that just today it was born his grandchildren. Child. Sorry.
Eva is her name. Thank you, Professor Zago. It's a pleasure to have you all here. I'd like to invite you to take your seats at the audience. I'm sorry, Professor Zago. Professor Cherry, you have the floor, please.
Thank you very much, Professor Zago, Professor Chi, Professor Hoff, Professor Pacheco, all the present. Well, I had the opportunity to work with Professor Brentani for many years. We had previously a familiar relationship. My mother and the family of Brentani were both from Trieste. So I knew Brentani before even entering at the University of São Paulo. And then, and I was at the Department of Radiology and the Oncology. Brentani decided to move to the Department of Radiology. Then we became the Department of Radiology and Oncology, something quite new, I think, all around the world. But we had a very successful relationship. I think we were a kind of complementary. I was more politic and Brentani more aggressive. So I think we did a very good job at the department. Then after that, when in my first term as dean of the medical school, he was an excellent collaborator, and he gave me many advice. And I think Brentani was an extraordinary person, very frank, very direct, and we were able to build a very strong department, and after, when he retired, we decided to create 2 chairs. One chair of clinical oncology that now Professor Paulo is the chair, and the chair of experimental oncology that Professor Roger Chamas is now the chair. It was a very good decision of having both branches, the clinical oncology and the research oncology in our department. And we were able also to build a very important cancer institute, the CESP. Now Paulo is the director, and also Brentani was very close. He at that time was retired, but he helped a lot with ideas in the creation of this institute that we are very happy to have as one of the most important hospitals in our complex in Hospital das Clínicas of School of Medicine. So I have to say that Brentani was not was not only a good friend, but a very important partner in the University of São Paulo and Medical School. So I'm very happy, very honored to be here, giving this tribute to a great researcher, a great person as Professor Brentani. Thank you very much.
Thank you, Professor Cherry. We now give the floor to Professor Paulo Hoff, General Director of the Câncer Institute of the State of São Paulo.
Thank you, Professor Zago, distinguished members of the table, friends that are here today. It is a great honor to be able to participate in such a meeting to honor Dr. Brentani. I was mentioning to Dr. Cherry, Professor Cherry, that it's incredible that 11 years have lapsed. It seems that it was yesterday. And contrary to some of you, I did not have the pleasure of working directly with Professor Brentani. Of course, I knew him well, and I think as being a representative of the State Cancer Center and also the president of Brazilian Society of Oncology, I can safely say that he has been one of the most important figures in the history of oncology in Brazil, especially being the first professor of oncology at the University of São Paulo, and now the advances that he was able to bring to the research of the oncology area here in our country. He was so active and such a powerful figure that we had to divide his chair in two to be able to do. And me and Roger, we have a great relationship, and I think that we do honor his legacy. And since I did not work directly with him, but of course participated in many meetings with him, I'll tell you just a brief story, and I think that shows well Dr. Brentani. I was organizing a meeting in Rio, and it was an international meeting, and we decided to talk, of course, about genomic sequencing, and all the rage was going on. This was 2002, 20 years ago. And I invited him to be the guest speaker and invited the president of the Chilean Society of Oncology, Dr. José Reyes from Clínica Las Condes in Santiago, to be the chair of the meeting. And he was very excited to meet Dr. Brentani. So he went to the computer at that time to study Brentani's bibliography and to be able to do a good introduction. And he spent something like 10 minutes talking about all the publications that Dr. Brentani had done and everything that he had done over the years, and invited Dr. Brentani. Brentani comes to the stage, thanks him, and says, "You know, I just have to tell you that you just introduced the work of my wife." So Reyes studied the wrong Brentani, meets his work. And he did this in such a way, and everybody was laughing, and he had great humor. And that shows that. So although I did not work with him, I guess I knew him well enough to be able to say safely that we all miss him, and I'm very thankful to be able to be in this table today. Thank you.
Thank you, Dr. Paulo. We now give the floor to Professor Carlos Américo Pacheco, CEO of FAPESP.
Thank you, Professor Zago, Professor Chi, Sergio, Paulo. It's a pleasure to be here. I have a few words. I met Ricardo Brentani a few times, Simpson also a few times. I was in the Ministry of Science and Technology in 1999, and I have the opportunity to meet him and to understand how sharp is the brain of Brentani. I will use the idea that his humor and his jokes, say some few words here in memory of Brentani. It is very hard for me to speak about Ricardo Brentani. Ricardo Brentani, as Professor Zago showed, was a brilliant scientist, and I am an economist. Most people that I know, all my friends, don't believe that economy is a science. So. Ricardo Brentani was a brilliant scientist and also an efficient administrator in the Ludwig Institute, in the Camargo, and also in FAPESP. At FAPESP, in the words of Carlos Vogt, his performance was passionate and critical. As his colleagues are unanimous to highlight his contribution to raising FAPESP level of quality, as Professor Zago just appointed, with his strong personality and sharp intelligence. Before Brentani, the role of the executive director of FAPESP, this position that I have now and Brentani had some years ago, was never very clear. Some people joke that the position was created to accommodate a friend of the governor. Ricardo Brentani gives a sense to this position. As appointed Erasmo Tolosa, who works with him at FAPESP, he never ran away from the fight. Brentani died when he was executive director of FAPESP. This is a big problem. Brito Cruz, our former scientific director, told my wife that all three of my predecessors died while in the office. Hopefully, the difference I have with Brentani will get me out of this one. Beyond the jokes, the important thing is to highlight that Brentani is an inspiration to all of us in terms of how FAPESP can do significant things when its board and its directors dedicate themselves with passion and critical vision to the formulation and implementation of relevant actions, when they go beyond the trivial and accept to risk and finance impeachables and disruptive things. Thank you.
Okay, I am very glad to introduce Dr. Chi Van Dang, who is the scientific director of the Ludwig Institute for Cancer Research, where his responsibility is to oversee the execution of the Ludwig's scientific strategy to advance the prevention, diagnosis, and treatment of cancer. He currently leads a Ludwig laboratory housed at the Wistar Institute in Philadelphia. I hope I'm right. John Hopkins now, so he's moving. Okay. And has held many positions in research laboratories, such as director of the Abramson Cancer Center at the University of Pennsylvania. He started his career at the Johns Hopkins University School of Medicine, where he was director of Division of Hematology, and eventually, I stress this because I'm a hematologist also, and eventually became the Johns Hopkins Family Professor in Oncology Research, the Vice Dean for Research and Director of the Hopkins Institute for Cell Engineering. He was the first to show that a master regulator of a gene expression named MYC, a gene whose mutation or aberrant expression is associated with many types of cancer, alter the metabolism of cancer cells. His presence here today is an opportunity to listen to one of the leading researchers in cancer, and also to commemorate the partnership between FAPESP and Ludwig in the FAPESP Genome Project. Please, Dr. Ching, thank you for coming here.
First of all, I want to thank Professor Zago and the leadership of FAPESP to have the honor to celebrate the life of Professor Brentani with the memorial lecture. Professors, ladies and gentlemen, it really is a great honor to be here, not only to celebrate the 60th anniversary of FAPESP, but also the Genome Project, and hopefully to also reinvigorate the relationship between FAPESP and so what I like to do is to give you a little bit of a historical perspective to celebrate Professor Brentani that you've already heard about. As you know, he was the scientific director starting in 1983 until his untimely death. So today, the Ludwig Institute actually has a number of sites throughout the world. So we have four branches around the world with several collaborating laboratories, including 6 centers in the US that have been endowed. The current strategy for the branches, which is funded by the Institute annually, is to really focus on the strengths of epigenetics, immunology, that is cancer immunology, and cancer immunometabolism to really bring new ideas into the clinic. So what I'm going to do is really give you a historical perspective of where cancer genomics started, celebrate what has happened here, interlace with this some of the current events, some of our own work, and then to really glimpse into the future of what are the promises that we will see. So let me take you back to 1911, the Journal of Experimental Medicine, when Peyton Rouse made a really interesting observation where a farmer came to him when he was at the Rockefeller Institute with chickens with tumors. And so he starts studying these tumors. What you can see from the family trees of the chickens is that it seems like after different generations, chickens get tumors. Those that are filled in dark are those who actually succumb to the tumor. And then there are a number of chickens that actually have spontaneous remission. And if you go into this paper very carefully, he presaged something today, which is immuno-oncology. Those that have spontaneous regression seems to have more lymphocyte infiltration. And this was really recognized in this paper, but he didn't really know what to make of that. So as you can see nowadays, we can't publish drawings of the tumors, but nonetheless, this was reported back then. And for this work, what he showed is that cell-free lysates are able to pass on the tumors, and this was really the discovery of the Rouse sarcoma virus. So for this work, he received the Nobel Prize in 1966. Then following his discovery, the Rouse sarcoma virus was partially purified, in vitro assays for transformation of normal cells to tumor cells, and the identification of the DNA provirus by Howard Temin, following by the isolation of the RNA. This is an RNA virus. And finally, the work that actually was seminal to really suggest that this transforming gene actually came from the normal genome, the so-called proto-oncogenes. And it turns out there was another type of virus called MC29, studied by other groups. And again, the paradigm held up is that you can find that the transforming agent, being the VMIC portion of the gene, in fact found its origin in cellular DNA. And this is work from the Bishop Laboratory. And as you can see, before all the modern technology we have, hybridization studies were done to demonstrate that in fact the cDNA of this particular retrovirus can hybridize with normal chicken RNA, demonstrating there's this gene that lies in the genome of the chicken. So of course for this work, Varmus and Bishop received the Nobel Prize in 1989. I had the fortune when I was a fellow at UCSF to actually work under the tutelage of both of them. And it turns out, what I want to take you back is in this era of pre-cancer genomics, that is the kind of work that, that has been contributed by the scientists here, is that there was genome science that was mostly driven by studying viruses. And in fact, the discovery of the Abelson oncogene came from studies of the murine leukemia virus. And in this case, the virus infects cells, capture Abelson, transform it into an oncogene. And later on, Abelson was found to be at the translocation breakpoint in chronic myelogenous leukemia. And in the far right, what I demonstrated is that today, patients actually benefit from this drug, Gleevec. Similarly, if you go back in history, you'll see that KRAS was discovered also from the Kirsten sarcoma virus. This is another murine leukemia virus that infects cells, capture this proto-oncogene and convert it into a transforming gene. And of course, just recently, a drug for KRAS was approved for lung cancer. So clearly, even in a pre-genomic era, there were just fundamental discoveries that actually can have clinical benefit. Now, the MYC oncogene itself, when it was discovered from VMIC, the human oncogene, was rapidly found in the 1980s to be translocated and activated in Burkitt's lymphoma, shown here with a child afflicted with this disease. And during this era, before the completion of the genome, we also have a number of seminal studies that suggest there are other genes that contribute to tumor genesis. This includes Al Knutson's seminal paper in 1971 in PNAS, where by purely studying the occurrence of retinoblastoma, he came up with this two-hit hypothesis. That is, he looked at children who manifest the disease at different ages and found that there are two different groups. One that actually have a very rapid onset, and the other one's delayed. And by modeling, he suggests that there's a double hit. Those that are rapid, that they have already inherited an abnormal copy of the gene, that is retinoblastoma, and then you have a somatic mutation in the remaining gene, whereas the ones that actually are delayed have to have two hits to get the tumor. So then following this was the study of retinoblastoma itself, and this is where a web cavity with Ray White was able to show and demonstrate and prove the two-hit hypothesis in this seminal paper on retinoblastoma. And as you know, web cavity was a director of the Ludwig San Diego for some time. It turns out that viruses also taught us about tumor suppressors, because SV40, the DNA virus, has a large T antigen that the retinoblastoma protein was found to co-precipitate it with large T antigen. This is work by David Livingston, who was who passed away last year, unfortunately, but he was part of the Ludwig Harvard Center. And then on the right, I just demonstrate that also the seminal work of a number of people, including Dr. Lane, who was also an institute director, found that P53 was bound to this oncogenes. In essence, SV40 large T antigen binding of these tumor suppressors inactivate them. And then the same year, in 1983, with the documentation of the retinoblastoma gene being, having 2 hits. Work out of Bob Weinberg's lab demonstrated in vitro that you can combine 2 oncogenes to transform essentially normal rat embryo fibroblasts to transform cancerous cells. And Bob is now currently director of the Ludwig MIT Center. And then the same year, in the same decade, work in the Vogelstein lab, particularly driven by Eric Farron, who actually after graduate school spent some years in my lab as a postdoc. They published this paper that also demonstrated multi-step tumorigenesis in colorectal tumorigenesis. It becomes known as a Vogelgram. And Bert is currently one of the directors of the Ludwig Hopkins Center. And then just taking you back, the Human Genome Project, this is all the pre-genomic era. It started in 1990, and you can see the timeline there. And you've heard quite a bit on when the first sequence was delivered in 2001. You heard quite a bit about the approaches on figuring out the Human Genome Project, including shotgun technique, and then how the role of ESTs play a role in actually identifying the coding sequences, as you've heard today. And then I just want to recap what you've heard already. This is a big celebration because it's a celebratory event to bring back in again the piece from Nature when it was announced that there will be a joint effort with FAPESP and Ludwig to sequence ESTs. And then on the right side, I show from The Economist a celebration that starts out by saying Samba, football, and genomics, something that is really to be proud of. And then, of course, you saw this as well in 2001, the Human Genome was published. And clearly, I just want to underscore the fact that the work from here really has made a major contribution in defining the transcriptome to be able to map out protein coding sequences, a great effort by the Ludwig Institute here. And then, of course, in a celebration mood, I just want to bring back, in 1997, it was announced and published in Nature about Brazil to sequence the first plant pathogen that you've already heard about as well. This is a picture that's shared with me by Andy showing a bunch of people working very hard to annotate the genome. And then, of course, on March 25th, 2000, it was a great celebratory event. And of note is that Andy was also a director of the Ludwig Institute here for some time. And as you can see, all the luminaries on the right there really was a proud moment for Brazil. Then, in 2006, the Cancer Genome Sequencing Project followed the completion of the human genome. This was effort not only in the US, but also there's an international cancer genome consortium. It was launched in the US in 2006. By 2010, 20 cancers were sought to be sequenced. And in 2018, the Pan-Cancer Atlas was accomplished with 10,000 tumors sequenced over 33 tumor types. So what have we learned from cancer genomics? And there are a series of papers in 2018 that report some of the lessons learned. On the right, I just illustrate a cartoon from a paper published in Cell in 2018 showing a number of lessons. For example, by looking at mutational spectrum of oncogenes, we learn about oncogenic signaling pathways. And here I just summarize some of the few key pathways, including NRF2, MYC, and P53. And then also, what's uncovered from this was mutations of epigenetic modifiers. And this is really something novel from the sequencing project. That is, mutations in epigenetic modifiers can then causes an alteration in the epigenome that then drive tumorigenesis, and also the role of non-coding RNAs that also emerges from all these efforts. So what has happened since then? Well, because of the Genome Project, we now, in many different medical centers around the world, particularly in the US, we use some of this genomic information to take care of patients. And this is just a cartoon from UCSF, which is really recapitulated in many different places, where one can build genomic testing from the cancer tissue of patients. Now, I was director of the Abramson Cancer Center. We actually created our own gene panels back in 2011, 2012. Once these genomic testings are done, clinicians then consult with the molecular tumor board to really begin to prioritize the availability of various types of targeted therapy, particularly kinase inhibitors. And this allows for a, if you will, a teaching feedback. And the system continues to enrich by teaching itself on what's best to do for patients using genomic sciences. And then, of course, the cancer genome also inform precision oncology through a number of different efforts in clinical trial design at NCI with umbrella and basket clinical trials, where you can take a particular cancer, molecularly profile it, and then compare different treatments to see whether the changes in the genome actually correlates with response. Conversely, one can look at basket trial, where you treat tumors by their molecular abnormality rather than the tissue of origin. And this is all the efforts that's really informed by genomic sciences. And then the opportunity that is also in front of us is prevention. So to understand germline risks, we can use genomics for interception. A good example of this, I just want to highlight, is the Bassler Center for BRCA that I'd helped launch when I was at Penn, owing to the donation from the Gray Foundation, who has a big interest in BRCA. And currently, I'm helping to lead an effort to create a pre-cancer atlas in breast and ovarian for BRCA. So basically, the idea is to take prophylactic surgical specimen and really to look at clues for the various very earliest lesions in these tissues using single-cell techniques, including transcriptomics, proteomics, and also single-cell imaging. And so this is ongoing effort, but this is really an area that is developing, and we hope that by, again, using genomics, that we're able to actually learn from all these data to be able to jump in and intercept, whether that's by pharmacological means or by immunological means, to really keep these patients and delay their prophylactic surgery if possible. Then, of course, the other area of opportunity is liquid biopsies. And this is a booming area. Bert Vogelstein has been a pioneer in this area. I just want to illustrate here, there's a number of different approaches, whether it's the circling free DNA epigenomics, work that's being done at the Oxford Ludwig branch. And then you also have fragment analysis, something that Victor Vogelescu is leading in at Hopkins. And you also have the canonical cell-free DNA genomics. And at the bottom, I just illustrate that, in fact, companies now are providing services so that you can have these, if you will, liquid biopsies to really help determine therapy. In this case, I just illustrate mutations that can advise the clinician on the various kinase inhibitors that could be used. And then I just want to turn a little bit onto MYC. This is an area that we've been working on for a long time. It turns out it's a oncogene that's frequently amplified in human cancers, shown on the left from CO Bioportal from the TCGA data set. On the right is a summary of the MYC pathway that's been shown in the pan-cancer analysis, where MYC itself is commonly increased, shown there in red, as well as NMYC, a family member that's well known to play a role in childhood neuroblastoma. And then in the shaded blue are some of the negative regulators in this network of MYC proteins. And so over the years, we've been very interested in the function of this gene, this oncogene, getting into functional genomics. And what we contributed to the understanding of its function is really to provide some of the very first evidence that MYC produces a protein that behaves like a transcription factor by first identifying the losing zipper in MYC that allows for the identification of MAX, its binding partner, also mapping out the transcriptional regulatory domain in the amino terminal end, leading to the concept that MYC and MAX are transcription factors that binds the DNA, and that's just a crystal structure shown on the right. And since that time, we've been very interested in studying the functional genome, that is, the transcriptome that responds to MYC activation, and work by many different laboratories, including our own. First, we contribute to the area of how MYC regulates cellular energetics, but then it turns out that MYC is able to turn on many different pathways that promote tumorigenesis, including immune evasion. And so our work in cancer metabolism really started out by the old-fashioned of doing subtraction cloning, looking at what MYC might turn on in a very simple experimental setting where the slide didn't actually show through on the right. Nonetheless, we identified several dozens of genes by subtraction cloning, and we decided to really, instead of studying the cell cycle, we decided to study lactate dehydrogenase A, which we demonstrated was a target of MYC. And this was the first time where an oncogenic pathway is linked to altered cancer metabolism, well known for many years, for a decade now, by work by Otto Warburg in Germany, showing that there's a propensity for many cancers to convert large amounts of glucose to lactate, known as the Warburg effect. And it's a nice book that was published a couple of years ago, really highlighting his life and the work. And my work, as probably all of you know, could not have happened without key trainees. And the two people who are pivotal to really transform our lab was Brian Lewis, my first graduate student. Hyun-Suk Shim was one of the very early postdoc. And it was really their work through subtraction cloning with all the older techniques, including slot blotting and RNase protection assay and nuclear run-on assays, to really document some of the MYC responsive genes. It turns out that the paper at the top, where we clone our RCL now, turns out that it is an encodes for a gene involved in nucleotide metabolism that's now recently shown to be synthetically lethal with PARP inhibitor for BRCA loss. And then Hyun-Suk Shim was really the champion of connecting MYC to LDHA, getting us into this cancer metabolism field. And we're very fortunate to be recognized by Nature as a milestone. That is a paper in 1997, and I heard before that was a magic year here as well. So over the years, we've contributed to the concept that MYC turns on many different metabolic pathways, and other laboratories contribute to the concept of proliferative metabolism. That is, when a cell is stimulated to grow either by normal signaling through growth factors or by oncogenic changes, whether it's receptor tyrosine kinases or activation of PI3 kinase, RAS, or MYC amplification, it turns out that these pathways can be boiled down to two key events. One is the activation of MYC, where it binds to MAX, to transcribe many messages that is required for cell growth and proliferation. And then activation of mTOR, that is also downstream of signaling pathways, including activation of amino acids. mTOR then allows for the translational amplification, that is, to take the messenger RNAs and make proteins. And many of these proteins are essential for bringing in nutrients into the cells to begin to build the building blocks for the cells to grow in size, prepare for DNA replication, and divide. So that's proliferative metabolism that can be deregulated by any oncogenic pathway. It turns out that non-proliferating cells have something that I label as maintenance metabolism, and it is largely driven by the circadian clock. So we all have circadian clocks that is really evolved to really contend with a day and night cycle because of our solar cycle, so that we undergo phases of feeding and fasting, waking and sleeping. Pretty much all of our cells have molecular clocks that causes an oscillation of the transcriptome at every 24 hours, driven by two hydrodynamic B-Malin clock. And it is a synchrony between individual cells' behavior and the organismal behavior of feeding and fasting cells that allows for the cells to synchronize the cells to be active during the day for us and undergo rest at nighttime, undergo repair, such as DNA repair. And what I show on the far right It's just some work that was done with my colleagues at Penn, where normal mouse livers were collected every 1 hour for 48 hours, and at each time point, 4 livers were harvested. And then you can perform metabolomics on these samples, and what you can see, not shown label, is that up and down on the y-axis are different metabolites. And as you can see in this heat map, red being high, green being low, is that over day and nighttime, you can see that the metabolome oscillates quite dramatically. That is, maintenance metabolism is not steady state. It actually fluctuates with the different metabolites to register with the organismal behavior. So the circadian clock, shown here, regulated by clock and BMAL, the way it functions as an oscillatory system is that it regulates 2 negative feedback loops. And as you know from engineering principles, that negative feedback loops allows for a system to be increased and then shut off and decrease again and to return back to a higher level. And so when clock and BMAL binds to target genes, many of these are metabolic genes, it causes a rhythmic maintenance of metabolism. So a number of years ago, we got a clue that gave us some insight to what MYC and MAX might do in terms of a transition from maintenance to proliferative metabolism. And what we recognize is that the clock and BMAL protein, which is crystallized, binds to a core hexamer on DNA that resembles very much like the core hexamer of MYC and MAX, which was also crystallized. So we hypothesized back in 2012 that many of the maintenance metabolic pathways are maintained by transcription factors that involve metabolism. But then when the cells are activated to grow and MYC levels are increased, then MYC can bind to very similar E-boxes, basically take over this so-called proliferative metabolism, so purely hypothetical. So then we hypothesized perhaps if when you have oncogenic MYC, when it's very high, that MYC can bind to clock and BMAL E-boxes, turn off the negative feedback loop to shut off the clock, and then to bind to target metabolic genes to turn on high levels of metabolism. And in fact, I won't show you all the data, but really the data supports the hypothesis. And I just want to illustrate to you that if you were to take a model system where you can turn MYC on and off and collect RNAs at various time points, you can see on the left, the transcriptome oscillates quite dramatically as a function of time when you collect samples from this experimental system every 4 hours over 2 days. Then when you turn the MYC on, you can see that the heat map of this oscillatory transcriptome is dramatically disrupted. Likewise, if you do metabolomics, you can see some oscillation of a number of different metabolites that, again, when you turn MYC on, it is disrupted. So we hypothesized perhaps oncogenic pathways could disrupt the circadian clock. Let me just summarize what we think happens is that in maintenance metabolism, all the pathways are ongoing to maintain the cells, but it's regulated primarily by the clock. But then in system where MYC is activated, MYC can turn off the clock and then activate proliferative metabolism to the transcriptome, heightening metabolism. And we found an opportunity to really translate this, because in early studies done by Han-Suk Shim, we found that cells that deregulate its expression in MYC, in fact, can undergo apoptosis when you withdraw glucose, because if the metabolic pathways are constitutively turned on, the cells become addicted to various nutrients, at least in vitro as shown here for glucose on the left. Then a laboratory at Cold Spring Harbor a number of years later showed that MYC-activated cells also are addicted to glutamine. And this led to the concepts that perhaps oncogenic pathway driving metabolic vulnerabilities can produce targets for therapy. And I won't go into the work that we've done in this area, but all these concepts have created a number of companies in the US targeting various components of metabolic pathways, and some of these are in actually clinical trials, and we're waiting for some of the more current ones to hopefully to see some successes. But one of the successes that should be celebrated was also from cancer genomics. And in 2008, Vogelstein's lab published an analysis, a sequence analysis of human glioblastoma multiforme, published in Science, and a little bit of story about Bert. When I was vice dean for research, sometime in 2002, I believe, Bert and Ken Kinsler came to my office and said, we need to borrow some money from the medical school. And when Bert asks you to borrow money, you don't say no. I just said, how much? He said, $2 million, because we got to sequence. And this was the paper. We got to do more sequencing, and it's very expensive. So without checking with the dean, I said yes. So then I went to the dean. I said, Bert needs $2 million. He said, he has it. But then it's on the books of the Cancer Center. So the Cancer Center director was not very happy with me because it shows as a red ink on the budget there. But nonetheless, the School of Medicine covered it. And really, the result of this is that the sequencing of GBM reveals one thing that's very interesting is the IDH, isocitrate dehydrogenase mutations. And following that, they then did more sequencing and found that this was quite pervasive and found in a significant fraction of GBMs, followed by the discovery of IDH mutation in, again, genomics, where Tim Lay sequenced the entire genome of a patient sample of AML. And in this particular case, they chose a case where there were no chromosomal aberrations. They were just looking for mutation. Again, they uncover IDH1 mutation. And in 2009, Agios Pharmaceutical discovered, in fact, these mutations causes a neomorphic function of the enzyme. It's not a loss of function, but it caused the enzyme to convert alpha-glutarate to 2-hydroxyglutarate, which is an oncometabolite. And from this discovery, they were able to produce a drug that is specific for the mutant form of IDH, and that was FDA approved for the treatment of AML with these mutations. So let me just, in the final moment, turn to an area that is quite active. It's really non-coding RNAs. We spoke a lot about coding RNAs. And in our work early on, over a decade ago, we were the first to demonstrate that actually in the MYC transcriptome, that it turns on microRNAs. And in particular work with Josh Mendel, his wife was a graduate student in my lab, was first author. We found that in fact MYC regulates a number of different microRNAs. We focus on miR-17 locus and found, as summarized in a cartoon, this MYC is able to activate this locus, which is oncogenic in itself. And it turns out it can activate E2F1 or inhibit P10. And subsequent to that, we demonstrate the connection between MYC and metabolism to the discovery that MYC can suppress miR-23A and B, which suppresses glutaminase. And so the world of non-coding RNAs came into our door. And we also pursue the area of long non-coding RNAs and identified some of the most potent responsive long non-coding RNAs is something termed dancer. We also showed that it also participates in MYC tumorigenesis. And finally, with the work that was done primarily at Penn, we did a whole genome analysis of long non-coding RNAs across human cancers. And then more recently, I just want to highlight, this just came out about a month ago, where in studying circadian transcriptome, we found a long-coding RNA called ADARF-AS1. And interestingly, we found it is a target of BMAL clock. But importantly, what we found is when we try to find out its function by using proteomics, we found that ADAR-AS1 in fact bound to this tumor suppressive complex, PBAF complex, a component known to be highly mutated in renal cell carcinomas. About 38% of cases, in fact, have mutations of PVRM1, a component of this complex. So when we found that it's bound to this complex, we asked the question, could it be that this long non-coding RNA is oncogenic by binding it to a tumor suppressor and inhibiting it? And so one of the clues we got by using TCGA data shown on the right is that if you look at only the expression of this long non-coding RNA is only predictive for survival only in the wild-type tumors, but not in the tumors that are mutant. Because if it is mutant, then this long-coding RNA does not need to inactivate this protein any longer. But it's just to highlight the fact that there are many more clues in the non-coding part of the genome that could really instruct it on tumor genesis. So let me just end with a couple of thoughts. Some of the opportunities in terms of genomics. So clearly, the area of therapeutic resistance is still a booming area to really understand. There's clearly, this is functional genomics. It's going to hopefully lead us to some clues on how to deal with this, whether we can intervene. A lot of work is now being done at the single cell level to look at all sorts of changes upon therapeutic pressure. The other area that is also a booming area of opportunity is single cell heterogeneity that can now be informed by single cell genomics as well as other omics approaches. And this is critically important because, as you know, tumor heterogeneity is really fundamental to therapy sensitivity or resistance. And finally, I just want to end with a couple of thoughts we heard a little bit earlier, is the cancer metagenomics. And it turns out, just by chance, I also highlight the same paper that was spoken about this morning, where the metagenomic analysis of colorectal cancer actually revealed a signature that's related to choline degradation. I just want to highlight a little bit about this. It turns out that in the diet, when we eat and food that contains choline, there's a specific enzyme, a microbial enzyme called CUT-C, that metabolizes choline to trimethylamine. And then the liver converts trimethylamine to trimethylamine oxide. That has been very well documented in the cardiovasculature as playing a role in cardiovascular diseases because it increases inflammation. And then a few years ago, a paper in Blood showed that the same pathway, if you trigger it, that is, you increase TMAO, you can also increase inflammation response and aggravate graft versus host disease in the model. And they showed that TMAO, the compound that's produced by the liver from the bacteria, actually can polarize macrophages from the M2 to M1, which then can trigger GVHD. And finally, a paper that I'm really glad to be co-author with my colleagues at the Wistar and also at Penn, where we just published recently demonstrating that this particular metabolite can drive polarization to M1 to really boost immune response checkpoint. And the graph that just shown survival in a pancreatic model, murine model, where if you combine checkpoint therapy with this metabolite, that you can enhance survival, as shown in the upper curves shown there as compared to controls. So I just want to conclude that the cancer genome, which we celebrate today, we can go from sequence to action. I went through a number of different concepts, including early detection, which I think is going to give us a cancer-free world in the foreseeable future if we can detect cancer early, eradicate it. We also can come up with drugs for frank tumor presentation with tumors. And then we can see that by prevention, we actually can decrease the burden of cancer. And I also illustrate a number of examples where there are many opportunities, including epigenetic modifiers being done in the clinic now. And so looking forward, I think the cancer microbiome is a big opportunity that we may play a role between Ludwig and FAPESP in the future. So thank you very much for your attention and the honor to really celebrate Dr. Brentani's legacy.
So in this final moment of our event, I'd like to start by thanking very much Marianne, Anamaria, Luis Eduardo, and our communications department for a superb job on bringing an excellent program on this celebration. I was considering, as just invited by Professor Zago to say a few words at the end, what I would say. And I think there's a dichotomy that we can consider. One is that we're celebrating the 20th birthday or 25th birthday of a very important program led by FAPESP, which changed the landscape of science in the state of São Paulo and in Brazil. But as we look to the past, we also may look to the future. And there are a number of students here, not only here in this audience, but in the video sessions. Actually, I'm not sure if it was this morning, I met two students, I think from Ribeirão Preto, that came here. They work on cancer research, and they're actually benefiting a lot from superb lectures and a number of very, very interesting and frontline research presentations. And so we both look back to the past, celebrating a major achievement, and we look forward to the knowledge and the ideas and the research activities that may be developed by those students. In a similar way, we heard a lot, both today and yesterday, on how to set an agenda for research programs that change the research landscape on an agency. And as we are currently discussing how to set the strategy for research activities at FAPESP, again, it's of great value for us to be reminded of how this was set, and most of all, then, to end my line of thought, the original proposal, actually, to catch, let's say, the government or the board idea was, well, we'll cure amarelinho, we'll cure a disease on citrus. The program didn't do that, and it took a while for that to be developed. But the program actually had many offsprings, and including, it started with a plant pathogen and went on to cancer and went on to many different topics. And most of all, great quality science was developed. So I think that's very insightful as we discuss our future activities. Once again, thank you very much for all the distinguished speakers, for all of the audience, and most of all, for our superb organizer team. Thank you.
Okay, so we come to the end. I wish to thank again all those who came to this meeting, those who have been seeing this conference by the internet. I wish to thank the researchers and the speakers, and also thank again all those who helped to organize this meeting, as Luis Melo already mentioned. And it was a pleasure to be part of this, see many friends. Again, Emmanuel, Sergio, Paulo, Giovanni, and the Simpson. So long time, Roger Chamas. I think we had 2 exciting days of conferences. We reviewed the histories of these 20 years or so, as Perry said. Not exactly 20, 22, 25. Teresa. But the world, we looked at what is going on at the moment, especially in 3 areas: infectious diseases agents, agriculture, and cancer. But the world changed very much in those years. Science and technology progressed. In the turn of the century, it was an extraordinary feat to sequence the 2 million bases per genome in 2 years. Today, this can be achieved in a few days, perhaps in 1 day or 2. We sequenced the complete genome of the SARS-CoV-2 virus of the first patient here in São Paulo in 2 days. What is more, we can now look at the genome of single cells instead of looking to the average of a billion cells. We can look directly to what happens to the proteins of those cells and the lipids and the sugars and the other small molecules. We can look at the metabolome. Thus, I forecast an exciting period of research in the next coming 20 years in this area. And for FAPESP, I think the message for us is very clear. Keep on supporting good projects, Luis. Second, take risks and aim high. Third, promote enduring collaborations with good partners, as we did in the past with Ludwig Institute for Cancer Research. So, see you again in the next year for the 2023 FAPESP Genomic Conference. Thank you.
Luis.